In this video, we will show how users can generate an analysis notebook from raw RNA-seq data using the BioJupies web server. So to start generating the notebook, we can go ahead and click on the Get Started button. Here, the user is required to specify what data they wish to analyze, and can choose between searching thousands of published and publicly available datasets, uploading their own gene expression data for analysis, or using an example dataset to learn how to generate notebooks. So for this demo, we'll click on Your Data. At this point, the user is required to specify the format of the RNA-seq data they are uploading, and can choose between uploading a gene expression table containing numeric gene counts with samples on the columns and genes on rows, or raw sequencing data in the FASTQ format. So we'll go ahead and click on Raw Sequencing Data. At this point, BioJupies provides a form which allows users to upload FASTQ files from their computer's file system directly to the web server for notebook generation. So we can go ahead and click on Choose Files. And in this case, we can select 6 FASTQ files from the computer's local storage. We can then go ahead and click on Upload. And at this point, BioJupies is taking advantage of the Sharon API to directly upload these 6 FASTQ files in parallel to an Amazon storage bucket. This process can take as little as a minute, depending on the size of the FASTQ files and depending on the speed of the internet. Once the upload has been completed, we can go ahead and click on Continue. Now the user is required to specify information concerning the FASTQ files themselves, including the organism from which the files were generated, and whether the sequencing experiment was performed using a single-end or a paired-end approach. So in our specific case, we can go ahead and click on the mouse and select the single-end sequencing method, indicating that the 6 FASTQ files will be individually aligned to the selected reference genome. We can then go ahead and click on Continue. And at this point, this information will be used by the Elysium API to launch RNA-seq alignment jobs on the Amazon cloud using Docker containers which run the Callisto RNA-seq quantification program. These Docker containers will then download the FASTQ files, estimate gene expression for each of these, and then save the results to a text file uploaded on the Google Cloud. This process may take up to 15 minutes depending on the size of the uploaded FASTQ file. Once the alignment step is complete, we can go ahead and click on Continue. The web server collects information from all the quantified results and combines them into a matrix of gene level counts, which is then previewed below. We can now click on Continue and proceed to the next step of notebook generation. Here, the user is required to input metadata describing the samples in the uploaded dataset. The user can either specify the group the samples belong to, using the form on the left, or upload a sample metadata file using the form on the right. So in this case, we'll click here and select the sample metadata file from the file system. In this case, we can then see that the first 3 samples in the dataset correspond to a wild-type group, and the remaining 3 samples belong to a knockdown group. We can click on Continue. And during this process, the data and metadata are packaged into an HDF5 file and uploaded to the cloud, where they're made available for notebook generation. Once this step is complete, you can proceed to video number 5 for more information on generating a notebook from your recently uploaded dataset.